am Dr. Naina Shirbhate. Hi friends, I am Dr. Naina Shirbhate, Assistant Professor in the Department of Botany, Vidya Vikas College, Samudrapur. In this section, we have discussed about the lipid metabolism. Lipids are the esters of fatty acid and glycerol, or it is a chemically diverse group of molecules that dissolve readily in organic solvent but only sparingly in water. It is an important constituent of cell structure and function. Due to their hydrophobic and non-polar nature, lipids are different from the rest of the other body compounds and unique in their action. It composed of fats and oil and composed of long chain of hydrocarbon, that is the fatty acids and glycerol. This is the structure of triglycerol. It is a type of one of the storage form of fatty acids. Hydrolysis of this fatty acid to form the glycerol, one molecule of glycerol and one molecule of free fatty acids. Nomenclature: fatty acids vary according to the length, hydrocarbon chain and number and position of carbon-carbon double bond. It is categorized into the two main types, that is the saturated fatty acid and another that is the unsaturated fatty acid. Saturated fatty acid having no double bond. These fats are saturated and semi-solid at room temperature. Example: stearic acid, palmitic acid. This stearic acid having the 18 carbon containing fatty acids. To see the structure of this fatty acids, here is the palmitic acid having the CH3C, CH2 by 14, CH3, COH. Now the unsaturated fatty acid, this fatty acid having the double bond, this fatty acid liquid at room temperature, and these are the oils are highly unsaturated. Example: linolenic acid, that is the 18:3 18 carbon compound fatty acid and having the 3 double bonds. To see, this is the structure of this acid. Now the role of fatty acid. Fatty acids are the concentrated fuel reserve of the body. It is important constituent of structure and regulate the membrane permeability. It serves as a source of fat soluble vitamin, that is the vitamin A, D, E, K. It protects the internal organ and serves as an insulating material and gives shape and smooth to the body. Other functions of the fatty acid: it is the structural component, intracellular storage depot of metabolic fuel. It acts as a protective covering, protect from the cell of many bacteria, leaves of higher plant, exoskeleton of skin, insect and skin of vertebrate. It acts as a regulatory substance. It transport form of neurotransmitter. It acts as a receptor in nerve ending membrane. It determines immunological specificity, and in case of enzyme it acts as a cofactor. Types of lipids are the four main types, that is the fatty acid. This fatty acid having saturated and unsaturated. Then glyceride, these are the glycerol containing lipids. Non-glyceride, these are the sphingol, steroid and waxes. Complex lipid, that is the lipoprotein, phospholipids and glycolipids. Structure of lipids: fatty acids attached to the glycerol through a ester link and form between the carboxyl group of fatty acid and hydroxyl group of the glycerol molecule. Here you see the fatty acid and glycerol are esterified by removing water molecule and form the triglycerol. To form the triglycerol, then localization of fatty acid found in animal as well as the plant cell. It is the structural component of plant cell membrane. Then plant and animal cell membrane is also found in the different organelles, covering of different organelles found in the some types of fatty acid found in the cytosol of cell. Now the fatty acid metabolism. Fatty acid biosynthesis mainly occur in case of stoma of chloroplast in case of plant and cytoplasm of animal. Then formation of acetyl-CoA, NADPH and ATP takes place at the time of fatty acid biosynthesis. Fat, at the time of fatty acid biosynthesis, acetyl-CoA and oxaloacetic acid combine in the mitochondria and form the citrate. This citrate passes to the cytoplasm with the help of various transporters present on the inner membrane of mitochondria. This citrate passes in the cytoplasm and break into the part of acetyl-CoA and oxaloacetic acid with the help of citrate lyase. This acetyl-CoA form fatty acid with the help of fatty acid synthase complex or machinery, that is the fatty acid synthase. This oxaloacetic acid converted into the pyruvate and returned back to the mitochondrial matrix to the transporter. This is the fatty acid biosynthesis. At the time of synthesis of fatty acid, acetyl-CoA carboxylase, which is present in the stoma of chloroplast. This chloroplast absorb carbon dioxide with the help of ATP. ATP is converted into ADP and inorganic phosphate. This carbon dioxide absorbed by acetyl-CoA. Later on, this acetyl-CoA converted into the malonyl-CoA. To see here, this malonyl-CoA later on converted to malonyl ACP. Again, this malonyl ACP is converted into beta ketoacyl ACP, later on into beta hydroxy ACP. Later on, this beta hydroxy ACP converted to enoyl ACP, acetyl ESOP, and in this way produce the phospholipid. This phospholipid, it is an important constituent of mini membrane. This fatty acid chain are produced in the stoma of chloroplast, and this fatty acid transferred to the cytoplasm for the various metabolic purposes. NADPase is needed for the purpose of reduction in the form of pentose phosphate pathway. ATP is also required for the synthesis of fatty acids. These are the differences of fatty acid biosynthesis and oxidation of fatty acid. Biosynthesis of fatty acid carried out in the chloroplast and oxidation in the peroxisomes. Then biosynthesis of fatty acid, for that carrier is the acetyl carrier protein, and oxidation CoA. Activation of CO2 required in case of biosynthesis and not required in oxidation purpose. Then in case of biosynthesis, add 2 carbon compound, and oxidation remove 2 carbon compound. NADH is required in case of biosynthesis and oxidation. In case of oxidation, NADH it generate. This biosynthesis of fatty acids require a multi-complex, that is the ACP synthase complex present in the stoma of chloroplast, and oxidation, it is the independent of enzyme. This is a summary of fatty acid biosynthesis. Not reverse, it is a 4-step process, that is the condensation, reduction, dehydration, and again reduction. It regulates in different animals. Important role of structural and biological functions in plant. Now the storage lipid. Storage lipid mostly found in the seeds and fruits. It's stored in the form of triglycerol and in the form of waxes also. It biosynthesizes from the glycerol and fatty acids. Fatty acids is the saturated and unsaturated. With the help of this glycerol are later on glycerol converted into the glycerol kinase in presence of glycerol converted into glycerol 3-phosphate in presence of enzyme glycerol kinase. This glycerol 3-phosphate converted into the lysophosphatidic acid in presence of enzyme G3-phosphate transferase. This lysophosphatidic acid later on converted into the phosphatidic acid in presence of enzyme acetyl transferase. This phosphatic acid lastly again converted into diacyl glycerol in presence of enzyme phosphatidic acid phosphatase. This acyl transferase diacyl glycerol is converted into the triacyl glycerol in presence of enzyme acyl transferase. In this way, this triglyceride is produced and stored in the seeds and other parts of the plant. Now the membrane lipid. Membrane lipids are different type. This is the structure of membrane. that is the phospholipid, glycolipids, sterols, archabacterial, ether lipids, etc. These membrane lipids synthesize in the smooth endoplasmic reticle and mitochondrial inner membrane, and this phosphatidic acid from the cytoplasm or plastid, they convert it into the membrane lipid. These are the complex forms of lipid. This is the phosphatidic acid. This converted into the 1,2-diacylglycerol or CDP diacylglycerol. Later on, this converted into phosphatidylcholine or phosphatidyl-enoyl, and these are the important constituent of many membranes. Now the oxidation of fatty acid or breakdown of fatty acid. Oxidation of fatty acids are different types. This is the alpha oxidation, beta oxidation, delta oxidation, etc. From these, beta oxidation is the most common. Then beta oxidation, proper beta oxidation, it occurs into 4 different phases, that is the oxidation, hydration, again oxidation and cleavage. Firstly, the acyl CoA dehydrogenase. Acyl CoA is converted in acetyl CoA, converted into the trans-enoyl CoA in presence of enzyme acetyl CoA dehydrogenase. Later on, this del2 trans-enoyl CoA converted into the beta hydroxy CoA in presence of enzyme beta hydroxy CoA transferase. Then this beta hydroxy CoA converted into the beta ketoacyl CoA. Beta ketoacyl CoA in presence of enzyme dehydrogenase. In this process, NAD is converted into NADH and H+. Lastly, the beta ketoacyl CoA converted into the acetyl CoA, acetyl CoA in presence of enzyme thiolase. In this way, the breakdown of fatty acid occur in the form of acetyl CoA. Odd number of saturated fatty acids such as palmitic acid produced in propionate. This is not a directly ester in the metabolism and other intermediate form. Now the functions, some important functions of this fatty acid is the energy reservoir, constituent of membrane, take part in various reaction, and some other function. It is the constituent of many membrane component, that is the suberin, cutin. It protect membrane from the various inhibitors. These are the references used, Leininger, Biochemistry and Molecular Biology book by Buchanan. Thank you very much.